Welcome. The DRSC Integrative Ortholog Prediction Tool, or DIOP for short, is an ortholog and paralog search tool that combines ortholog predictions from several different algorithms. A paper describing DIOP was published by Hu and colleagues in 2011 in BMC Bioinformatics. Since then, we have made additions, changes, and improvements. This video is based on version 6.0.2 of DIOP, which was released in June 2017. DIOP supports ortholog and paralog searches for most common model organisms and human genes. To do an ortholog search, choose the input and output species, or choose an input species and select all to view results from all other species. To do a paralog search, choose the same species as the input and output species. In this video, we will discuss how DIOP ortholog pair scores are calculated and how terms like rank, best score, and best score reverse are assigned to a given ortholog pair. The first important point to know about DIOP scores, ranks, and best calls is that they do not provide a definitive answer as to whether a given pair of genes is the true orthologs or paralogs, however those terms are defined. You should consider the scores, ranks, and best calls predictors or indicators of the likelihood of an orthologous or paralogous relationship, not definitive answers, and plan to do further computational and/or experimental analysis. The second important point to know about DIOP scores, ranks, and best calls is this. Scores are based on the number of algorithms or other resources integrated at DIOP that predict a given ortholog pair. Rank, best score, and best score reverse incorporate this voting system DIOP score, as well as other information. To see what this looks like in practice, let's look at what inputs on the search page contribute to the DIOP score. We will begin with an ortholog search for zebrafish orthologs of the human Wnt1 gene. You might notice that when we select Danio rareo or zebrafish as the output species, some of the algorithms listed on the right-hand side disappear and ZFin appears. The algorithms that disappear do not cover zebrafish, so they will not be counted in the DIOP score. The ZFin resource has ortholog mapping information for zebrafish versus human, so it appears when we select zebrafish. This leads to the third important point to know about DIOP scores. Because not all algorithms and resources integrated at DIOP cover all the species we support, the highest score depends on which species are selected as the input and output species. Let's look at the results page. You will quickly see that one of the predicted ortholog pairs, human Wnt1 versus zebrafish Wnt1, has a DIOP score of 12. On the right-hand side, you can see the specific 12 algorithms or resources that predict that human Wnt1 and zebrafish Wnt1 are orthologs. You might also notice that the other results shown have scores of 2, and the 2 algorithms or resources that predict these pairs are shown. Next to the score is a weighted score. The weighted score is adjusted based on the calculation of the predictive value of each resource, as described in more detail in our 2011 publication and on the documentation page. Let's go back to the search page. Here you have the option to exclude scores less than 1 or less than 2, or to select only the best score. We again see the human zebrafish Wnt1 pair, but now the other results are not displayed. You can see that this pair is marked as the best score pair. That simply means that no other ortholog pair has a higher score. If 2 pairs share the highest score, both will be marked as best score. It is also marked as the best score in the reverse direction. That means that if you started the search with zebrafish Wnt1 as the input and human as the output species, the same pair would be the highest scoring pair. You will also see that this ortholog pair has a rank of high. This example follows the general rules we apply to assign ranks. The orthologous pairs with the best scores for both forward or reverse searches and that have a score of 2 or better are ranked as high. Pairs with the best score for either forward or reverse search but not both and that have a score of 2 or better are ranked as moderate. Pairs that do not have the best score for either forward or reverse search but have a score of 4 or better are also ranked as moderate. Everything else is ranked low. Now let's try the same search but select all species as the output. Notice that when we do this with the return best score filter on, DIOP displays the best score matches for each of the species included. Let's try the search one more time, this time selecting human as the output as well as the input species. With this type of search, we can view putative Wnt1 paralogs in the human genome. Like our first ortholog search, you will see that several predicted paralog pairs are displayed. Each is assigned a rank, score, and weighted score, and whether this is the best score and best reverse score is also displayed. Let's start over with a query that looks for human orthologs of the Drosophila wingless gene, which has a gene symbol WG. In the results, we see that 11 algorithms or resources integrated at DIOP predict an ortholog relationship between fly wingless and human Wnt1. This pair also has the best score, would give the best score if the reverse search had been done, and has a rank of high. Looking down the list, you can see that some other pairs have a ranking of moderate or low. It is important to look at the reverse search information. In this example, Wnt4, Wnt3, and Wnt3a have the best score with a reverse search and a rank of moderate. These genes are potentially good candidates for additional or human orthologs of the Drosophila wingless gene. Wnt ligands provide a well-studied example of genes with paralogs and orthologs in common model systems. We invite you to use examples you are closely familiar with to get a sense of what the scores and rankings provide. This should help inform your interpretation of scores and ranks for your experimental genes of interest. You might also notice that some algorithms included at DIOP seem better to you given your understanding of your system and your goals as compared with others. Note that at the search page, in addition to the option of filtering by score or best match, you can also include or exclude any subset of sources of ortholog and paralog predictions. Here are the summarizing key points. 1. DIOP scores are not definitive. They are indicators of confidence. Further analysis and experimentation are needed to define true orthologs or paralogs. 2. DIOP scores are based on account of how many algorithms or resources integrated at DIOP predict a given ortholog or paralog pair. 3. The highest possible DIOP score would depend on the input and output species selected, as not all algorithms and resources integrated at DIOP cover all the species we support. We welcome your input on the DIOP resource or other online resources from the DRSC, which can be viewed at fgr.hms.harvard.edu. On the Online Tools Overview page, you will find a link to our bug report form. We try hard to respond to suggestions. This video was made in response to a suggestion submitted through the bug report form. We note that additional information about DIOP is available in our original publication on DIOP, in our Nucleic Acids Research 2017 database issue publication, and on the DIOP documentation page. We also note that DIOP scores and ranks show up in searches with our Gene to Function online resource, which is designed to give you a quick overview of functional information for orthologs of a gene of interest, as well as provide access to that information. This resource can be found at genetofunction.org. If your use of DIOP ends up contributing to a publication, please cite our paper and provide the URL in your methods section. This helps us continue to get funding essential for maintaining, supporting, and updating the resource. Thank you.